Good morning, everyone. Or, sorry, good day. It's morning when I'm recording this, so that's why I said that. I am going to record basically my direction on chromatography. Chromatography is a technique that we're going to do a couple of times this semester. We're doing ion exchange chromatography in lab this week. So we'll get started. Chromatography is a separation technique based on the different interactions of compounds with two phases, the mobile phase and the stationary phase, as the compounds travel through a supporting medium. The components of chromatography are the mobile, stationary phases, and the supporting medium. So the mobile phase is a solvent that flows through the supporting medium. The stationary phase is a layer or coating on the supporting medium that interacts with the analytes. The supporting medium on which the stationary phase is bound or coated. So we're going to be doing ion exchange chromatography. And so our supporting medium is something an insoluble sugar, something like cellulose. And then the stationary phase is ions that are basically permanently affixed to that supporting medium. The mobile phase is whatever buffer or protein containing solution that we're going to run through the column. Now, proteins first have to be harvested, and that's what we did the previous week. So the entire cell is harvested. If protein is intracellular, we have to break those cells open. And what we did was we used a blender and we used a pH 7 buffer to do that. In some instances, you can simply use the culture medium if the protein is exported. But if that protein is found inside the cell, you have to burst the cells open. So basically, this is asking the question, or this is addressing, where is your protein? If it's inside the cell, well, then you have to rupture that cell. If it's exported or if it's outside the cell, well, then you can just collect the medium in which that is grown in. Now, what we did for separating the components of our extract, so after we blended it up, um, we did protein precipitation. That was our ammonium sulfate precipitation. We used ammonium sulfate, NH4, and then parentheses 2, SO4. So sulfur or sulfate is a -2 ion. I apologize. Ammonium is a +2 ion. Um, in addition to protein precipitation, you can filter samples and separate your protein from other stuff. For instance, if you have a soluble protein, something that's just freely floating around in the cytosol, you can filter out any membranous material. And you can filter that out with a filter that looks not too dissimilar from a coffee filter, in which case you're talking about membrane filtration, microfiltration, or ultrafiltration. Another way of separating, however, is centrifugation, which is one of the steps that we did in our purification. Now, this last part that we will do is known as dialysis. Dialysis relies on the chemical concept of equilibrium. Dialysis is intended to basically remove something from a mixture. Now, let me use it here. So in our protein sample, if you remember, we had so we have the pellet, the ammonium sulfate pellet that we basically vortex to make that soluble. So we've got a bunch of ammonium ions and sulfate ions floating around in here. We've also got a bunch of proteins in here. I'm not really going to distinguish colors here or anything like that, but this is a solution with protein, ammonium, sulfate ions, a bunch of stuff in here, more or less. And some of those things we want to get rid of. Specifically, we want to get rid of the ammonium ions and sulfate ions. How would we do that? Well, one way that we do that is through a process known as dialysis. Dialysis uses a tubing that a couple of things about it is one, it looks like a snake skin, and two, it looks a lot like or when working with it, it's kind of like a produce bag at the grocery store, where it's like such a thin material that the two sheets of it, you can't even really distinguish between the two of them. So opening up that produce bag is kind of difficult. Much like opening up dialysis tubing is kind of difficult. Um, at any rate, what we do is here's our sample, and let's go ahead and just call this 5 milliliters, give or take some. What we're going to do is we're going to get some dialysis tubing. This dialysis tubing, we're going to put a clamp on one end because it's going to be open on both ends. So we're going to clamp it off. And then we're going to open it up and we're going to transfer all of this 5 milliliter sample into that dialysis tubing. We're going to clamp the other side off. And so it really is going to look to be basically sealed off and closed. But here's where dialysis tubing is super important. Dialysis tubing has extremely fine pores, extremely small holes in them. And dialysis tubing, these small holes, they have a cutoff that's known as an MWCO, molecular weight cutoff, meaning that these holes are of a certain size so that things can freely go into and out of this dialysis tubing. So if you saw an MWCO of approximately 10K, that means 10 kilodaltons. Now, if you remember, a dalton is 1 dalton, we'll say. 1 dalton, not 1 daltons. 1 dalton is the same as 1 atomic mass unit. Now, if we were to say something is 10 kilodaltons, that means it's approximately 10,000 daltons. You could also say that it's 10,000 AMU. And then a more practical way of looking at it is you could say that it's 10,000 grams per mole, provided that you have a mole of that substance. It's 10,000 grams. Now, if you only have one of those, it'd be 10,000 atomic mass units. Now, with that in mind, we have this tubing that has this cutoff. Things that are smaller than 10,000 grams per mole will enter and exit without a problem. So if we think about NH4, 2, SO4, my question is ammonium sulfate smaller or larger than 10,000 grams per mole? It's definitely smaller than, I'm going to call it 10 kilodaltons, KDA. So what that means is ammonium sulfate can enter and exit this tubing. Okay. Now, what we want to do is we want to get that ammonium sulfate and anything else that's smaller than 10,000 daltons out of this tubing. So we've transferred our protein sample, X, into this tubing. And so a little bit more, or different representation of it looks like this. Okay. Now, this is really built on the concept of equilibrium. So what we need to do is we need to find a 1 liter beaker and put our Na-phos buffer in here. So we're going to get that 1 liter beaker with the the sodium phosphate buffer, we're going to take this dialysis tubing that has our protein that has a bunch of ammonium sulfate in there, and we're going to throw this in here. Now I'm going to adjust my scaling a little bit. And what's slowly but surely going to happen is that this buffer out here, the sodium phosphate, that has no ammonium sulfate. Whereas inside my dialysis tubing, there's tons of ammonium sulfate. So what's going to happen is the ammonium sulfate is going to leave this tubing and enter this outer solution so that ultimately we can reach an equilibrium. And we can say, well, there's, you know, I don't know what that equilibrium would be, but there's going to be some ammonium sulfate in tubing and some, not zero. Oh, darn it. Some ammonium sulfate in large beaker. We are relying on the sample entering and exit, that ammonium sulfate entering and exit that tubing, and anything else that's 10,000 Daltons or smaller entering and exiting. We're going to replace that buffer a couple of times so that that ammonium sulfate can slowly but surely be diluted out of the tubing. Now, in time, what's going to happen is we are going to see other things exit that tubing. Do we have to worry about our protein leaving that tubing? No, we don't, because our protein is larger than 10,000 Daltons. So dialysis is what we're doing in order to dilute out ammonium sulfate from that 65% pellet. You do not have to worry about that dialysis. We will take care of that. Now, that's one separation technique, and that's really good for cleaning up a sample. Separating components, well, differences in proteins allow the separation of target protein from other proteins. Chromatography allows the sorting of proteins based on size or by how they cling to or dissolve in various substances. One type of chromatography, size exclusion chromatography. This is also known as gel filtration or GF filtration. This uses beads, gel beads with pores. Larger proteins move quickly around the beads and smaller proteins slip through the pores and therefore move more slowly through the beads. So the way I've always kind of thought about this is if you were to take I don't have a good example here in my office, but imagine that you had like a toilet paper roll or something like that. And the toilet paper roll has an opening on it has an opening on each end. And I'm going to draw that cylinder right here. And if you were to try to put a tennis ball on top of that, that tennis ball is not going to enter that tubing. And so what's going to happen is that tennis ball is akin to our large protein. Sorry, the toilet paper roll is the gel bead with the pore in it. The tennis ball is a large protein. Let's say that we've got a tennis ball, and then we've got an M&M, and we've got a golf ball. So MM, golf ball, TB for tennis ball. Okay. This tennis ball, if you put this and tried to make this go through this tubing, it's going to sit on the top. And in time, what's going to happen is that tennis ball, that large protein, is going to move on from that tubing. It's just going to go around it because it can't enter that tubing, so it can't do anything with it. Alternatively, the golf ball. Well, the golf ball is going to enter and it's going to go through, and it's going to go through a little bit more slowly than the tennis ball because the tennis ball could not enter that tubing at all. And so it just sort of hits the rim and moves on. The golf ball is going to kind of squeeze its way in there and then slide ever so slowly through there. And in the process, contacts all internal surfaces. Okay. Now, so what that means is the tennis ball moves through the tube, or sorry, it moves through the column, which has a bunch of these tubes in it, these gel beads. It moves through the column first. So large protein, or TB, exits column first. Okay. Now the golf ball, GB. The golf ball exits second. This last one, the M&M. Now, most folks are going to look at this and they're going to have this visual. They're going to be able to put these components together and say, okay, well, I've got a toilet paper roll, I've got a tennis ball, I've got an M&M. That M&M, it's certainly not too large. And so it won't have a problem going through there. But where it gets a little bit tricky where it gets a little bit tricky is this M&M. It's going to enter this tube and it's going to come in contact with the entire, or with this gel bead. And what it's going to do is it's basically going to spin around and 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 around until it exits. It's going to make contact with every single surface of this tube, just like the golf ball did. But if you think about the golf ball just had to kind of skate through and make contact, whereas the M&M, its movement through the tube is like this. So it has to make all of these rotations in order to contact every surface of the internal, every surface of the interior of the of that tube there. So this smallest protein, it exits last because it is riding through, it is making contact with every single point of the interior of that tube. So that is kind of an explanation for gel filtration chromatography. Largest proteins exit first, smallest proteins exit last, and the why. Okay. Now, when it comes to gel filtration or size exclusion chromatography, there's different types of materials that are used for gel filtration. For example, here we have, and these are just kind of you have your three big materials. Dextran, it is a homopolysaccharide of glucose residues, excuse me, generally used to separate small peptides, or generally used to for small peptide separation as well as small to average proteins. Polyacrylamide. These gels are prepared by cross-linking acrylamide with N,N-methylene bisacrylamide. Your separation ability varies by cross-linking degree. So how much these molecules, how much this acrylamide is linked in this network, that's going to influence the separation. And then agarose, linear polymers of D-galactose and 3-anhydro-1-galactose. This is best for larger molecules. What I want you to know is the three names, dextran, polyacrylamide, and agarose, and what they're best for. Dextran is for smaller proteins. Polyacrylamide, well, polyacrylamide, you've got a pretty good range there, but agarose is for your largest proteins. Now, if we look at these down below on this table, we've got dextran polyacrylamide as the green ones and agarose with the group at the bottom. Agarose, you're separating 10 kilodalton proteins to 4,000. That last agarose, you're separating proteins ranging from 70 kilodaltons all the way up to 40,000 kilodaltons. So I want you to think about this as if you're trying to separate a 15 kilodalton protein from a 75 kilodalton protein. Okay, so 15 and 75. I'm going to write this out. You have a mixture of two proteins. One is 15 kilodaltons and one is 75 kilodaltons. Okay, so your 75 kilodalton protein and your 15 kilodalton protein, you personally have no way to detect their difference. Well, 75, that might look good at the Cephros 2B, this one at the bottom here. But that would actually not be, like, agarose would be a bad selection for these proteins. The reason for that is this is going to be able to separate a 200 from a 500 kilodalton protein. But that fine tuning is going to be difficult. And so you're not going to get that good resolution and that good separation. Whereas a 15 and a 75, I mean, really, either dextran or polyacrylamide would be good options. I would lean towards polyacrylamide just because if you look at the ranges of some of these dextran columns, like these first three, 0.05 to 0.7, we're talking about very, very, very fine differences. That's not to say that this separation is outside of its capabilities, but think about it like, let's see. I'm trying to come up with a good analogy here. Like that dextran, that Cefadex G10 is great for this very, very fine separation. Whereas polyacrylamide is better for kind of a broader scale separation. You could always think about it like when you're adjusting a microscope or when you're looking through a microscope, you have the coarse dial and then you have the fine dial. The coarse dial is like it's going to move the either the platform or the optic up here to here or like these very huge movements, whereas the fine tune dial is going to move the optical feature this much. You're not even really going to notice it. And essentially what you're doing there is you're making these very fine minor adjustments compared to a large adjustment. So this is effectively what you want to look for. And so anyway, when we look at these ranges, be thoughtful about, okay, what is this column capable of and what is best, or what is it best used for? So I think that if I was separating a 15 and a 75 kilodalton protein from one another, the column that I would use is this Biogel P100 because I have a separation range from 5 to 100 kilodaltons, or it's best to separate something that's between 5 and 100 kilodaltons. Now, as a visual for what protein purification looks like, here is at the bottom here, this is what is known as a chromatogram. This chromatogram has two axes that are important. Your y-axis is some way of detecting your protein, and your x-axis is your either fraction number or time. And this can be time or it can be fraction number, especially because of the fact that if you're trying to collect, 10 fractions a minute, or if your column has a flow rate of 10 fractions per minute, you'd be able to extrapolate and convert that between time and fraction number. So what this is showing is at the top, this table, or part A and part B, these two things are interconnected with one another. In column A, or sorry, the very first representation of your column, you have your proteins in a mixture where there's been no separation. They basically are loaded onto the top of the column, and they're going to begin to move through that column. Your small proteins are going to kind of congregate at the top because they're moving through that column very slowly, whereas your large proteins are moving through it a little bit more quickly. Those large proteins will move through quickly or fastest, and those are going to be the ones that elute first. So those are going to be within your first few fractions. Your next group of proteins, or your next kind of batch of proteins that are going to come out of the column, are your smaller proteins. They're going to end up distributed in the latter few of the columns, or later as the column continues to run. Now, the type of chromatography that we're going to be doing in lab is ion exchange chromatography. This relies on the charge of a protein. Resin is charged, or the stationary phase is charged. Opposite charged proteins will stick to resin beads, can be eluted by changing the charge with salts or increasing in concentration. So here's a representation of what that is. If you look first and foremost at the chromatogram at the bottom, we have a y-axis that is, well, we actually kind of have two y-axes. One of them is your A280. That's a pretty standard way to detect protein. The other is your concentration. So concentration related to the elutant. So whatever solution elutes protein from the column. Okay. Now, if we look at this, what we've got is a couple of different proteins. Or sorry, the first thing that I want you to recognize about this is this line right here, this kind of salmon colored line. It's going to be low and effectively zero. But then right here, you see the slope increase, and it goes up in a pretty linear fashion. And then it stops right here. Okay. Now, let's look at the black lines. The black lines correspond to, or the black lines have nice little peaks here, here, and here. Those correspond to a star, a darkened circle, and then another salmon colored circle, or salmon colored dot. Those correspond to our different proteins. Okay, so let's jump up to the top here. We've got our mixture of our different proteins. In our column, where none of our proteins have begun to find their space, we have a bunch of positive charges. That's because our resin, or our column bed, is positively charged. These proteins up here will begin moving through the column. And depending on their charge, if they are negatively charged, they'll hang around and they'll bind to that column because our column has a positive charge. If the proteins are positively charged, by contrast, they're going to move through the column because they're going to, if anything, be repelled by that column. So we have phase one, where our proteins have not entered the column. Phase two, where our proteins have entered the column and they're going to begin flowing through, and those uncharged proteins, or those positively charged proteins, which have no ability to interact with our resin. Those positively charged proteins are going to go straight through the column. They're going to, when our elutant buffer is got a concentration of 0, those proteins are going to elute from that column. Okay. Third phase is proteins with a slightly negative charge. These proteins are going to correspond here. So they have a negative charge and they will adhere to the column. And in order for them to come off the column, we have to have some buffer concentration that goes up to get those proteins off of that column. Now, the next and last one is these kind of salmon-colored proteins, which they are only going to elute if the salt concentration is this high. Now, the numbers are not provided here or anything like that, but let's go ahead and say that right here is 0.1 molar, and then right here is 0.3 molar. Now, with that in mind, the protein Y elutes with 0.1 molar, and I'll just say NaCl. Protein Z elutes with 0.2 molar NaCl. Now, what's important to recognize is that these proteins, these respective proteins, will elute with a concentration of 0.1 molar or higher. So protein Y, it will elute with 0.1 molar. It will also elute with 0.2 molar. Protein Z will not elute with 0.1 molar, but will elute with 0.2 molar. So if you were trying to isolate protein Y, what concentration of sodium chloride would you use to elute that protein? You'd probably want to use a 0.1 molar solution, because protein Y will come off the column, protein Z will not. Oh, I apologize. So that's important to recognize with respect to like separating proteins from one another. Protein X will come off the column without any sort of interference or intervention. I mean, you just use the wash buffer with no salt in that column. That protein's going to come off the column. Okay, so with that in mind, these are the different substances or the different resins for ion exchange chromatography columns. And basically the groups that you will find on them. So DEAE, cellulose is what it's commonly called. And that's diethylaminoethyl cellulose. So that's a sugar bead, an insoluble sugar bead, but this group right here has been added to it. And so that is going to attract anions, because if you look at the group, it's got positive here, positive here. It's going to attract anions. Meanwhile, if we go down here to SP, we've got a negative charge right there. That's going to attract a cation. So it's these beads, these sugar beads, insoluble sugar beads, have X. And that X is this functional group right here that has been added to it. Now, as you change the pH, that group may or may not be deprotonated. If you look at something like Q, well, that has a range from 1 to 14. That's fantastic. That is always going to work as an anion exchanger. Whereas something like diethylaminoethyl, DEAE, has a pH range from 2 to 9. What I want you to know about these different groups is I want you to know about something as a strong or a weak anion or cation exchanger. If it's positively charged, then it's going to attract negatively charged ions. If it's negatively charged, it's going to attract positively charged ions. Those would be the most beneficial things to understand about this. Now, affinity chromatography relies on the ability of proteins to bind specifically and reversibly to uniquely shaped compounds called ligands. So affinity chromatography is a technique that lots of people tend to think like, oh, that must be the the gold standard. Like this is perfect because if you have a substrate for your enzyme, you just get that substrate and you permanently affix it to those beads and you're going to be able to capture your protein. And it is very good to think about this technique in that way. But, you know, if you think about trying to purify hexokinase, excuse me, or trying to purify a protein that binds to a hexose, there's lots of proteins that do that. So I think when it comes to affinity chromatography, I think that it's very good to do ion exchange chromatography, salting out, any sort of filtration, all of these things leading up to affinity chromatography, using affinity chromatography or gel filtration chromatography as your last step, because then you've kind of had these filtration steps, or these steps where you basically say, I'm not worried about these non-specific things, not, and you get rid of 70% of the junk, and then you're left with 30% of the proteins that can be your protein of interest. Okay, so then one last chromatography technique is hydrophobic interaction chromatography, sorts proteins on the basis of their repulsion of water. I hope I'm recording. Yeah, good. And as you would imagine, we're dealing with the hydrophobicity of a ligand, and the hydrophobicity of a protein. If it's very hydrophobic, then it's going to adhere to this. If it's not hydrophobic, then it's not going to. So proteins have hydrophobic regions, and they will be attracted to different parts of this column. Then I'll go ahead and stop right there with the PowerPoint. I'm discarding my ink. And now what I want to do is talk to y'all about the actually no, I gotta run back. Sorry. So this step right here has at the bottom of it I mean, this is not too different from what we previously talked about. But this is a figure that shows the migration and separation of proteins through a column. At the bottom, we have a chromatogram, and that chromatogram has detection, a way of detecting a protein, your detector signal, and then on your x-axis, time. So what we're looking at is the time that it takes for these proteins A and B to separate from one another. A elutes first, B elutes second. Now, this could be representative, or this is probably representative of a gel filtration chromatography column, because all that you're waiting for with gel filtration is time. You just have to give your sample time to migrate through the column. Now, when it comes to affinity chromatography or ion exchange chromatography, you have to worry about getting those proteins to elute from the column. If you look at your y-axis here, what we're looking at is we're looking at salt concentration. Now, these two different chromatography or elution techniques, these are a stepwise elution and a gradient elution column. So if we look at this column, let's look at the one on the left-hand side. First thing that we're going to do is we're going to just run some junk through, or run our buffer through our column and equilibrate it. This 5 to 10 CV is 5 to 10 column volume. So you get a sense of how tall your column is and what volume solution can go in there. And you say, okay, well, it has a 1 CV. This is a hypothetical column here. One column has a volume of approximately 2.5 milliliters. I'm going to run 25 milliliters of buffer through this column to basically equilibrate it. After I do that, then I'm going to take my sample, my protein mixture, my proteins A plus B plus C, I'm going to load them onto this column. And what I'm going to do is I still have a buffer, an elution buffer with a concentration of 0 molar NaCl. Okay, so I'm going to load my sample onto this column. No sodium chloride. I'm going to let the basically observe to see if anything comes out of the column. And sure enough, there's some stuff that comes out. And then what I'm going to do is I'm going to begin very gradually increasing the salt concentration. And what I'm going to see is as I increase my elution buffers NaCl concentration, I'm going to see it creep up. My sodium chloride concentration is going to be creeping up. And what I'm going to notice is that some proteins elute from this. Okay, great. That's great. I want to see proteins elute from this column. And what I've done is I've taken a mixer, a solution that can, or a instrument that can mix two buffers. And it can mix them perfectly where, one sample is, it's 1% buffer 1 and then 99% buffer 2. And then it's going to mix these buffers and it's going to mix and it's going to be 2% buffer 1 and 98% buffer 2. That's how we get this gradual increase in salt concentration. Now, the type of elution that we're going to be doing is the one that's seen over here to the right. Well, the equilibration of 5 to 10 column volumes is basically the same. The addition of your sample, however, that's where things are going to be similar, but things are also going to change a little bit, because what we're going to let happen here is we're going to collect all of these unbound molecules. Then we're going to very abruptly switch to a buffer that has a considerably higher salt concentration. So let's say that buffer, I'm going to jump over to the left. Buffer 1 has a concentration of 1 molar NaCl. Buffer 2 is 0 molar NaCl. In this next column, we're going to have a buffer 1. I'm going to call it buffer 1.5 and a buffer 2. So buffer 1 is 1 molar NaCl. Buffer 1.5 is 0. Just a second. Did this backwards. Just a second, sorry. Okay. Buffer 1 is 1 molar NaCl. Buffer 1.5 is 0.5 molar NaCl. .25 is going to be 0.25 molar NaCl. And then buffer 2 is going to be 0 molar NaCl. Okay, so I'm going to erase some of my previous markings, and I'm going to write in a different color where each buffer is going to be used. I'm going to write it in blue. So we're going to start out by equilibrating the column with buffer number 2, where you have no NaCl present. Then we're going to make a fairly abrupt jump, where we're going to use buffer number 1.25, where we have some NaCl. Okay, and what we see is that there's some proteins that elute out with that 0.25 molar NaCl. Then we're going to make the jump from buffer 1.25 to buffer 1.5 up here. The salt concentration is 0.5 molar NaCl. And then last but not least, buffer number 1. Buffer number 1, we're going to see our NaCl concentration go all the way up to 1 molar. So what we've done is we've made these fairly big jumps. So from 0 to 0.25 to 0.5 to 1 molar NaCl. Those jumps in our NaCl concentration is going to help us elute different proteins. So if a protein elutes with a 0.25 molar NaCl solution, it's going to be found here. If a protein elutes with 1 molar NaCl, it's not going to be here. It will not elute. So we're just washing away proteins that elute with a lower concentration. If something requires 0.5 molar NaCl, well then it's not going to be mixed with the thing that elutes with 0.25 molar NaCl. So the big difference here is a gradient elution and a stepwise elution. A gradient elution, I think, is very helpful when you're trying to explore and you're trying to figure out, oh, what concentration will help this protein elute? A stepwise elution is a helpful elution when you know what you're trying to separate. Well, I know that my protein elutes with 0.25 molar NaCl, and a bunch of junk elutes or co-elutes after a concentration of 0.4 molar NaCl. So it's helpful to have this information and know the difference between a gradient elution and a stepwise elution. Stepwise has big steps, big jumps in sodium chloride concentrations, whereas gradient elution is a very gradual increase in salt concentration. Okay, now I'm going to discard that. Now what I'm going to do is close that, go to the experiment. Okay, and so here we are. All right. So ion exchange chromatography, there's a bunch of questions about these different techniques or about the different columns. The protocol. You're going to be collecting some data from this. You're going to be measuring absorbance at 280 nanometers. That's in the UV spectrum, so you need to know to use a UV cuvette. Your IAs will have those UV cuvettes set aside for you to use so that you won't use the wrong one. There are differences, and if you use the wrong cuvette, you're not going to get the kind of optimal reading. Now, what we're first going to do is we're going to load a protein mixture. We're going to use the 15,000 RPM sample and load that onto your column. So everyone will be purifying lactate dehydrogenase from the 15,000 RPM sample. Very similar to what we previously prepared. Everyone will have approximately 1 milliliter of this. You're just going to use your milliliter or your fraction and load that directly onto the column. So whatever you set aside from last week, load all of that onto your column. Now, what you're going to be doing is essentially, I'm going to switch a share to whiteboard. There we go. What you're going to be doing is you're going to first load sample to column. You're going to load your sample onto the column, and then what you're going to be doing is you're going to be washing the sample or the column and collecting fractions. As soon as your sample is put onto the column, begin collecting fractions and collect them in microcentrifuge tubes, and then read the A280. You're going to read your A280 of all of those fractions. And what you're going to watch and what you're going to observe is that number is going to go up, and then it's going to go down. And at some point, it should hopefully get close to zero. And whenever it gets close to zero, then you'll go ahead and begin, as it gets close to zero, then you're going to switch buffers. That elution buffer, you're going to switch to a different elution buffer, and you're going to switch to 0. I believe that there's a total of 5 different elution buffers, and you're just going to repeat this process. I believe that the first one is a 0.02 molar NaCl, and you're going to again see that NaCl solution have your A280 go up, and then it's going to go down. And when it gets close to zero, you're going to switch to a new buffer. I believe the next one is 0.05 molar NaCl. And you're going to go through this process where you're using this buffer, watching the A280 go up, come down. Go to the next buffer, A280 goes up, comes down. Go to the next buffer, A280 goes up, comes down. You're going to do this several times. Collect and save all those fractions, record your A280 for all of those, because your A280 tells you whether or not there's protein in a fraction. You're going to collect all of these fractions, save them, because we will use them whenever we're doing catalytic assays. With all of that said, your instructional assistants will be able to help you with kind of getting a sense of, oh, should I move on to my next buffer? I would say that you'll, at the end of this, have at least 25 fractions total. So what that amounts for is about 4 to 5 fractions from each buffer, including the initial sodium phosphate buffer. There's no need for anyone to have like 60 fractions. If you've been collecting fractions with your first buffer, and after 10 of them, your absorbance hasn't gone back down to 0, I would change buffers. I would move on to the next buffer. So basically putting a limit of no more than 10 per buffer, but not too fewer than 5 per buffer. So 5 to 10 for each of the different buffers. Okay. I hope that was helpful, and I hope you have a good one. All right. Bye.